PI3 kinase pathway. PI3 kinase pathway is activated by RAS, just like the MAP kinase pathway. The signaling molecule will activate RTK, which then activates GRB2, which activates SOS, which activates RAS. RAS goes on to activate PI3 kinase. PI3 kinase goes on to activate PIP3, and PIP3 will activate PDK1. PDK1 will go on to partially activate AKT, and mTOR complex 2 will fully activate AKT. And AKT will go on to activate effector proteins downstream of this pathway. The first initial part of signaling molecule activating RTK activating GRB2, GRB2 activating SOS, and SOS activating RAS is something that we covered previously, and that will remain unchanged for this pathway. We're going to look initially at PI3 kinase activating PIP3. Now here we have activated RAS and an activated receptor tyrosine kinase with signaling molecule bound. And we have PI3 kinase in the cytosol, and PI3 kinase will bind to the activated RAS and itself become activated. Once activated, it will bind to the activated receptor tyrosine kinase. This is activation through localization because now PI3 kinase is near its substrate. What does PI3 kinase do? PI3 kinase is an enzyme that catalyzes the following reaction. Phosphatidylinositol 4,5-diphosphate PIP2 plus ATP will give us phosphatidylinositol 3,4,5-triphosphate PIP3. And this is the reaction that is being catalyzed by PI3 kinase. The part that's highlighted in the picture below is part of the PKC pathway, PIP2 to IP3 plus DAG, which is catalyzed by PLC beta. Proteins with a pH domain can bind to PIP3. Green fluorescent protein, GFP, will emit green light under the right conditions. The GFP gene can be fused to DNA sequences of other proteins, leading to the production of GFP-fused proteins. Under a fluorescent microscope, these GFP fusion proteins can be visualized in a live cell. In the cell picture to the right, cells were either given the gene for GFP or a GFP pH domain fusion protein. C and D are negative controls because you have a GFP fused to a protein with a deleted pH domain. So essentially the GFP does not have a pH domain. The arrows are showing you the nuclei of these cells, since the GFP fused protein is cytosolic and should not appear in the nucleus. The cells were treated with epidermal growth factor or not treated. Epidermal growth factor will activate the RAS pathway, including the PI3 kinase pathway. Activated PI3 kinase will produce PIP3. In cells that were exposed to EGF, the GFP pH domain fusion protein localized to the cell membrane because you have PIP3 now on the cell membrane, and the GFP pH domain fusion protein will bind to those PIP3 molecules in the cell membrane through the pH domain. So here in this picture you can see that PI3 kinase that has been localized to the cell membrane goes on to activate PIP3, meaning that you're increasing the concentration of PIP3 in the cell membrane by catalyzing the reaction of PIP2 to PIP3. PDK1 has a pH domain and is localized to the cell membrane by binding to PIP3. Localization of PDK1 to the membrane is its activation step. Next is the activation of AKT. AKT has a pH domain and is localized to the cell membrane by binding to PIP3. AKT is now brought near PDK1, and PDK1 will partially activate AKT by phosphorylating it. mTOR complex 2, which is activated by a separate pathway, will phosphorylate and fully activate AKT. AKT is now fully active and will dissociate from PIP3 and activate downstream effector proteins such as BAD. Activation of PI3 kinase pathway leads to many responses by cells. Some important examples are aiding in cell survival, stimulating cell proliferation, stimulating cell growth by increasing protein synthesis, angiogenesis, and cell motility. In non-growing cells lacking growth factor stimulation, the intracellular levels of PIP3 are kept very low by a series of phosphatases. One such phosphatase is PTEN, which removes the 3' phosphate from PIP3 which was previously attached by PI3 kinase. So PIP3 to PIP2 catalyzed by PTEN is the off switch. PIP2 to PIP3 catalyzed by PI3 kinase is the on switch. The pathway involving PI3 kinase and AKT is deregulated in a number of human cancers. In advanced ovarian carcinomas, deregulation of PI3 kinase is associated with increased proliferation and invasiveness as well as resistance to apoptosis. PI3 kinase is hyperactivated in almost 1/3 of human colon carcinomas. In a number of distinct tumor types, including breast and prostate carcinomas, PTEN activity is lost. Overall, PTEN activity is lost in 30 to 40% of all human cancers. So here we're showing PTEN and how it inhibits the PI3 kinase pathway.